Hello students, I am Dr. Ann Maxton, and today we will be understanding the principle, working and the components and their functions of a light microscope. I would like to acknowledge Dr. Raghuveer Singh for assisting me and helping me arranging and in compilation of the different materials required for the practical. I would like to acknowledge his effort. Students, I will begin with the light microscope. Let us understand. Firstly, let us understand the contribution of the scientist, that is Robert Hooke. The use of microscopes for studying cells is called basically a microscope. The first microscope was designed by Robert Hooke in 1665. The microscopes are instruments for making enlarged images of ordinary invisible objects which are not visible to the naked eye. The microscopic objects are measured by units such as microns and angstroms. The primitive microscope used by Robert Hooke students was very simple and consisted of only a combination of magnifying lenses fixed in a tube. Compound microscope has a combination of lenses which comprises concave and the convex lenses. Now, the microscope that you are seeing right now is an optical instrument which is used to magnify the objects, and the magnification of an object by an optical instrument means that the size of the retinal image, the image formed on the retina of our eye of the object under the view, is increased. The ratio of magnified image to that formed on the retina of an unaided eye is called magnifying power or magnification of that particular instrument. A microscope has two lenses, the ocular at the top of the body tube and the objective lens at the end of the body tube, close to the object to be examined. Both these lenses have magnifying power. There are different types of microscopes, students. Based on the source of light, microscopes are of two types. They are light microscope and electron microscope. The microscope in which ordinary light is used as the source of illumination is said to be light microscope. The light microscopes are again of two types. They are simple microscopes and compound microscopes. Simple microscope, a light microscope with two lenses, is said to be a simple microscope. It has only low magnification power. Example, the dissection microscope. Whereas the compound microscope is a light microscope with more than two lenses, and that is the reason why it is known as a compound microscope. Since the compound microscope has more than two lenses, the total magnification is effected in more than two times. A typical compound microscope consists of strong metal body or stand composed of a base and an arm to which the remaining parts are attached. Talking about the parts of a microscope, students, let us understand the different parts of a microscope. The microscope that you are seeing right here is an instrument used to see objects that are too small to be seen by the naked eye. The microscope in science of investigating small objects and structures using an instrument. Okay, this technique is known as microscopy. And the microscopic means invisible to the unaided eye. Okay, that is the microscopic view that is required to see the objects that cannot be visible by the naked eye. The optical microscope also referred to a light microscope. Light because you see the illumination source as the light. Now, this is a type of microscope that is commonly uses visible light and a system of lenses to generate magnifying images of small objects. Okay, so let us see the parts of a microscope. Here you see the eyepiece. Number 1, you see the eyepiece. Now this is the eyepiece, the ocular lens, also known as the ocular lens. The eyepiece or ocular lens is a cylindrical, it's like a cylinder containing 2 or more lenses. Its function is to bring the image into focus for the eye. The eyepiece is inserted into the top of the end of the body tube. This is the body tube. The objective, turret, resolver or resolving nose piece. Now this here is known as the revolving nose piece. Revolving. Why? Because it can revolve. This is the revolving nose piece. It is also known as the objective turret, revolver or revolving nose piece. As we see in our physical body, that the nose is the part of the face on which the lenses that we put are resting. Similarly, the magnifying lenses that you are seeing here, they are resting upon the revolving nose piece. Now this nose piece is helping to hold multiple objective lenses. Objective lenses are supported by the revolving nose piece, and you can set whichever lens magnification you want. Here we see that the objective lenses are there. At the lower end of a typical microscope that you see, there are 1 or more objective lenses. Here you see 1, 2, 3 objective lenses of 10x, 10x, 100x and 40x magnification. You see 40 here, 40, 100 and 10. So there are 3 objective lenses of different magnifications that are 10x, 40x and 100x. Now there is a coarse adjustment knob. Here you see, students, a coarse adjustment knob, and you see a fine adjustment knob. A fine adjustment knob. Okay. Now this is a very important thing to note. They are maintaining the distance between, what is this? This is the stage. This is the stage. These are the stage clips. These are the stage clips. And to move these stage clips, you have 2 knobs. Okay. I repeat, and I go back from the top and I will come down with the parts. Remember, I spoke about the ocular lens, the eyepiece, the body tube, then here revolving nose piece, objective lenses. This is your neck, known as neck, and this is your handle, and this is your base. This is the base or the resting platform of the microscope. This is the wire that supplies the electric current. You need to switch on and off the electric supply to function the microscope. Then there is a switch right here. You can switch off and on the electric supply. Okay. Now here is a coarse adjustment knob and a fine adjustment knob. Coarse adjustment knob will alter the distance between the stage on which you place your slide. For example, you have a clean slide. Let us take a clean slide and place it on the stage. Once you place it on the stage, you need to see that the slide in the place where the sample is should be exactly under the light and the objective lens. Okay, so you can move left and right, forward and backward to adjust the sample or placed on the slide. Coarse adjustment knob will help you for the first stage of focusing. once you focus and you visualize the object from the ocular lens, you use the fine adjustment knob to make it even more clear and to set the resolution correct, so that the image is not only magnified but it is very correctly and finely viewed through the ocular lens with your eye. Here you find the condenser. This is the condenser. Now condenser plays a very important role. The condenser plays a very important role. The role of the condenser is that it condenses or focuses the light beam, the source of light. See here, the mirror, okay? The source of light, the light beam is focused and condensed with the help of the condenser, so it strikes straight through the object to the objective lens, through the condenser, through the object, and then through the objective lens, all through the body tube, all magnified image to the retina. This view is captured by your retinal lens. Okay, so this is all the parts of the microscope. Before I end, I repeat. The parts of a microscope: ocular lens, eyepiece, body tube, neck, handle, base, electric supply switch, coarse adjustment knob, fine adjustment knob, stage clips, diaphragm and condenser, light source and the base. Thank you very much, students. I hope you have understood the parts of a microscope and their functioning and the principle behind a microscope. Thank you.